all. This is Dr. Mobeen Sayed from Drbeen.com. Welcome to one more show. Today we're going to talk about the Pfizer's vaccine a little more. So first of all, we're going to talk about the news that the FDA's advisory committee has approved or recommended the approval of the vaccine today. The voting was 77% versus, I think, no or absentees or abstinence. Now there is the second step is for CDC to approve it. Their advisory committee meeting is on this Sunday. So hopefully, I'm not exactly sure what is the process for these two individual organizations, but looks like FDA will be the next one to approve it. Then the CDC on Sunday will have their advisory committee meeting and voting, and then they would approve it, hopefully, and then it will be fully approved maybe, and that is how we'll move forward. So this is a great news. I know that some of the cool beans are upset with me that I am celebrating it, but this is a great news. Here's the reason. Let's say that there are some cool beans who do not believe in vaccination, and they think there is something evil going on with the vaccination as well. There is still a majority of the people who will get vaccinated. I am in those people. And that would create herd immunity. That would protect from possibility of death in many who are at risk. That would reduce misery. That would allow us to start opening up our society after the vaccination. So there is a lot of silver lining in here, even if the whole population does not participate in it. I'm not going to sit here and try to convince someone who is not convinced for vaccine. Maybe so the sound bites that I've heard or the written bites that I've seen are that, hey, it is too rapidly approved. There is no animal studies in it. It is approved in a very short time. And so because of that, it is not tested properly. Or this is a new kind of a vaccine. So we do not know exactly the kind of side effects that may appear, or what will happen to autoimmune diseases, or what if somebody already had the COVID-19 and then they have the vaccine. So there is a long list of possible doubts and concerns, and these are valid concerns. It's not that we have answers to all of these things. Although you would see that today I would have answers to some of these things. So let's start a discussion. So quick soundbite. FDA committee has approved it. Next step is on this Sunday for the CDC committee, and hopefully we'll start giving the vaccine, administering the vaccine after that. Now, what I wanted to do was, so this is the whole discussion for today, but what I wanted to do was, I wanted to go over the safety and efficacy document, which I had shared a couple of days ago, and actually look at a few things that are very important. One of them is, what are the ingredients of this vaccine, the one that Pfizer has said. And if Pfizer has given a list of ingredients and then there is something else in it, Pfizer can really be in trouble. So I wanted to go over those ingredients with you and kind of make sense of what these ingredients mean and what are the possible ingredients in there that may cause allergic reactions. Number one. Number two, I wanted to look at the safety data for not only the healthy people who got the vaccine, but those folks who had autoimmune diseases or had HIV or had SARS-CoV-2 or the COVID-19 before, and then they were given the vaccine as well, or were younger in their age. So I thought that all of that data is present in that document, and I wanted to go over that with you, so that you can either develop confidence or at least answer some of your questions from there as well. So if you are comfortable with reviewing a long document with me, I have gone over it. I have underlined it and highlighted it. So I think it will be fast, but still, if you're comfortable doing that, then I'll invite you to let's look at that document together. What do you think? Should we do it? Okay, let's start. So the answer to the questions that we are going to see here are Number one, there was a question by someone today on Twitter that the vaccine contains the viral spikes in it. So, technically, literally, no, but it does contain the RNA for the spikes, which, when goes into our cell, will help make the spikes, which are then presented on the cell surface in a controlled fashion, either on MHC1 or on MHC2, and then immune system is triggered to respond to that antigen and to learn to fight with that antigen in the future. So I hope that that answers that question. We talked about it yesterday. We talked about it when we talked about Pfizer's vaccine a couple of days ago as well. The other thing that I would request you to keep an eye on is the Bell's palsy. So there were some folks who had developed Bell's palsy uh, in the placebo, in the vaccine trial, in the vaccinated group. And I would like us to look at that as well, because there are some people who are concerned about it. So with this, let's start. Is there a question? Good question, John. I am too. So what was the question? So I'm curious what they made the liposome from. So lipids. So let's look at it. Let's look at what they disclose in the document itself. So Bell's palsy is the drooping of the one side of the face, or kind of paralysis of the side of the face. It did occur in 4 people, but they all recovered. It was temporary, and the incidence rate was the same as general population's Bell's palsy rate. Okay, so I'm going to start now. So Jude, yes, I wanted to make sure that we talk about it. So here is the document. And so today, less drawing and more going through the document, so that if you wanted to go over it as well, at least there is a map of the document in front of you and you can feel comfortable. So if you see here, if you see in here, there is the vaccine composition and dosing regime. There is also another point here, which is about the vaccine ingredients. So let's continue. We'll find them as I have highlighted these things. So I'm not going to go over the intros, and just going to start with the data itself. So this is the first one. I guess this is the biggest part of the discussion today. If I was a little bit more commercially, I should have a clickbaity link here, title that said vaccine ingredients. I think that would have gone in millions. So check this out. The ingredients for the vaccine are: the Pfizer BioNTech COVID-19 vaccine is a white to off-white, sterile, preservative free. So this is important. Preservative free, because sometimes preservatives in the vaccines can cause reactions as well. Frozen suspension for intramuscular injection. Now remember, this is not coming from BioNTech Pfizer. It is a FDA document that I think they must have gotten help from Pfizer's consultants, but this is FDA's seal on it. The vaccine contains a nucleoside modified messenger RNA. So that is the mRNA. That is the primary thing that they are going to inject in us. So why not I do this? I'm going to kind of develop this thing in front of us here. So one, they're saying we have an mRNA. So that is fine. We are aware of it. This is the mRNA, which will help make the spike proteins. Then they say the vaccine also includes ingredients, lipids. So in that lipid, the specific lipid type is 4-hydroxybutyl azenadyl bis hexane 6,1-dial bis 2-hexyl D canoate. And then there is another which is 2-polyethylene glycol and di-tetra decadecyl acetamide. Then there is another which is 1,2-disteroyl SN glycerol 3-phosphocholine, and then cholesterol. Out of these, the phosphocholine is actually in our lungs as well. Cholesterol is in us as well. So these are just simple um, lipids. So what are these lipids? These are the ingredients, lipids in which this messenger RNA is enveloped. And how do they do that? What they do is they charge them differently. So let's say the messenger RNA is charged negative. Then they will make the lipids as positive or relatively positive. So that would cause the lipids to stick with the messenger RNA, and that becomes a lipid particle. So I'm looking at the comments as such here as well. So Janet says deltoid in arm, ouch. Yes, so there is some ouch here, of course. So this is the lipid particle. So these are the two primary ingredients. Then potassium chloride, so KCl. So there is some potassium chloride. Why is that over there? To maintain the pH of the substance. Otherwise, the things will start decaying. So there is a specific pH that needs to be maintained. Monobasic potassium phosphate, another salt, sodium chloride, another salt, dibasic sodium phosphate dihydrate, and sucrose. So some salts, if I can just say here, salts. And then it's not necessary that they are inside. I'm making them inside the particle. KCl here, more salts here. Imagine that this vaccine is sitting in a bottle. And then sucrose. Sucrose is sugar, correct? So just our sugar, the sugar that we eat. So sugar here. And then, of course, they would have water as well. So let me just put some water here. Salt water, normal salt water, sodium chloride water. So this is the vaccine. Now, the reason for sugar in there, and this is not a concern for diabetes, it is a very tiny amount. The reason for the sugar in there is that when the vaccine is frozen, the sugar molecules will prevent various lipid particles to stick with each other and become one solid unit. So these kind of keep boundary between the particles. So now we have lipid. This lipid particle is possible that it can cause allergy. So the folks who developed allergies, out of all of these ingredients, they possibly developed allergies to the lipids that are present here. mRNA is enveloped in it, so it is not going to cause allergies. KCl or potassium chloride or sodium chloride or other salts are not going to cause allergies. Sucrose or sugar is not going to cause allergies. And there is no preservative to cause any allergy. I don't think there is any egg substances or fetal tissue things or adenovirus in here. There is no other thing other than this. So hopefully this answers the question on the ingredient side and kind of makes you comfortable with it. The Pfizer-BioNTech COVID-19 vaccine is supplied as frozen between -80 to -60 degrees centigrades, multi-dose 5-dose vial. The vaccine must be thawed and diluted in its original vial with 1.8 milliliter of sterile 0.9% sodium chloride injection. So what they would do is that when they'll take the vaccine out, so first they would thaw it, they would kind of make it less frozen. After thawing that, then they would inject the 0.9% sodium chloride, the one that I made here. They would inject that water in it, and then as they would dissolve it and they would shake it and they would mix it, then they'll take a dose out. So that water is not going to harm it. It is fine. This water is present in many, many things. And the reason for the water 0.9% sodium chloride injection is to make the vaccine in balance with our tissue fluid concentrations. So our tissue fluid are at the same 0.9% kind of sodium chloride concentration. So this just to match the vaccine with our tissue fluids. After dilution, the vial contains 5 doses of 0.3 milliliter per dose. After dilution, the multiple dose vial must be stored between 2 to 25 and used within 6 hours from the time of dilution. So you take a frozen vaccine out, you thaw it. In that thawing process, it can live for 5 days, but you thaw it as soon as it is thawed. Then you inject water in it, or 0.9% sodium chloride, not exactly water, but a sodium chloride solution. You inject that, it becomes 5 doses, and then within 6 hours you use and apply those doses, administer those doses. The Pfizer BioNTech COVID-19 vaccine, called BTN162b2, 30 microgram, is administered intramuscularly as a series of 2 30 microgram doses, 0.3 milliliter each, 21 days apart. So Pfizer is a messenger RNA vaccine. Moderna is a messenger RNA vaccine. I believe there are other as well. If I think about Oxford vaccine, AstraZeneca, that is a adenovirus vaccine. I believe Sinovac is adenovirus vaccine as well. But these guys are messenger RNA vaccines. So I'm going to look at the questions now just to see if there are ingredient related questions. So James Kelly has a question. What is the specific lipid phosphodiethylenedicolorine? Isn't this needed into and can Pfizer fail to disclose and other adjuvants? Of course, James, for example, I know about phosphatidylcholine. This is surfactant in our tissue fluid as well. This is a normal lipid solution that is present in our body as well. So that is what they are disclosing. This is what they are disclosing here. And they are actually giving exact chemical formulas as well for the lipids that they have used. And the good news is it is right in front of us. And if they don't do it, then they are at fault. And that can be a problem for them. Janine says, I don't trust the vaccine, so I'll pass on this one. Okay. Any other ingredient related question? So Robin says, please talk about whether people with chronic Lyme's disease and co-infections like fibromyalgia and other autoimmune illnesses are in danger of reactions from any of the vaccines. So let me answer this question right now. They have actually given the vaccine to people with chronic immune diseases and autoimmune diseases and did not see any issues with the vaccine. So that is a good news. Sepal Steve says that the virus is rapidly mutating. Hopefully the vaccine still works. So the virus has been mutating from the very first day and rapidly mutating. And we know that if you go to nextgen.org, you would see so many millions of mutations. If I can actually very quickly do that. Nextstrain.org, I believe is the name, this one. And if you go here for COVID, the main SARS-CoV-2 page, I hope this is the one. Look at the strains. All these dots are its strains. And if you play this, you would see that this is such a, look at just observe the window now, the way it is playing, and you can speed it up and you can slow it down. And these are all mutations. These are all variants that are found. But still, with all these variants, with all these mutations, the spike protein is kept consistent by the virus because that is the hand of the virus and it has to use it to bind to ACE2. So fortunate for us that it has not found a way to change this spike protein and make it something else, or allow it to bind to some other receptor by creating a different kind of a binding protein. So we are good. So your concern is correct, but I would say that so far this spike protein has, even with so many mutations, it has not mutated. So we should be okay. This is possible that when the vaccines come in and a large swath of people have become immune to it, and the virus is now just selectively becoming copies and the new copies are changing itself, maybe there is a new strain at that time for the selection pressure to occur, and or due to the selection pressure that some new strain comes up which has a successfully modified spike protein, and then we'll have to change the vaccine. That is something we do not know yet. So your concern is correct, but so far the mutation does not show that this would possibly happen fast. So I'm going to go back here. Let's look at it. Okay, so there is a question, Morris. Could the allergy reaction have been from histamine release during the immunization development? So what happened was the lipid in this vaccine reacted with the mast cell. So probably these folks mast cell who were hyperallergen already or hyperallergic already, their mast cell were already primed with some sort of a reaction to small antigens like lipid parts of the lipid, and they reacted to it. So this is why the folks who have severe allergies, that they have to keep the EpiPens with them or adrenaline with them or some other form of injection with them, they would need to be careful about this. Others, so far you would see that in here, you would see people with autoimmune diseases, you would see people who had COVID-19 before and then they got the vaccine, and you would see that nothing happened. Margaret says Lyme connections. So far, Margaret, Lyme as itself is not mentioned, but chronic illnesses and chronic immune illnesses and autoimmune diseases have been discussed, and they say that nothing, no concerns. I'm going to try to stay with this discussion here. I have responded to this question before as well. How long immunity will last, doctor? And why not to give one dose so more people get these doses available? Number one dose, I'll show you the efficacy. This part of the question is important, and I would answer that today. That what are the chances of getting the infection after one dose? And they have given that data. And then how long the immunity would last. Looking at previous SARS-CoVs, I've always said 2 to 3 years. But because these companies and FDA does not have this data, they would just always say, we don't know. So how did they get the RNA? That was a very, very simple one, Vincent. What they did was the genome for the virus, the genetic materials makeup, was released by China, I believe, in December, or not December, in January. Then the scientists in the whole world now, especially, advanced countries, they have been looking at the genome by themselves as well and looking at the strains too. So it is very easy to pick up the spike protein's genome. And from there, they created a copy of that genome, and that becomes the RNA that they would use. Okay, so I'm going to go back to the discussion here, long document. So this is the ingredients. So I hope that the ingredient question is answered, and the curiosity that what causes the allergic reaction, the lipid part of the particles can cause a reaction. The particle size itself is in nanometers as well, just the similar size as the virus itself. Okay, so continuing. And I'm going to go to those parts which I have highlighted. So in phase 2 and 3, participants were enrolled with stratification by age, so grouping by age. Younger adults 18 through 55, then 55 years and over. But if you see here, 12 through 15 years of age, 16 through 55 year of age, and 55 and older were also included. Although in the younger ages, they have given them vaccines, but they do not have sufficient data to come back and say vaccine protects or does not protect, because younger adults usually tend to not get the disease that often. So in the surveillance time, in the time when they were looking at people, if they are getting the infection or not, they couldn't collect enough data to say, yes, it helps, or no, it does not help. But they have given the vaccine to younger population as well. This has been a very common question too. Then look at this. This is an important paragraph. The study population for phase 2-3 included participants at higher risk of acquiring COVID-19 and at higher risk of severe COVID-19 disease, such as participants working in the healthcare field. Look at this: participants with autoimmune diseases, such as and participants with chronic but stable medical conditions such as hypertension, asthma, diabetes, and infections with HIV. I have received a lots of questions about HIV and can they receive the vaccine or not. So here is that answer. Hepatitis B, hepatitis C. So these folks were given the vaccine and nothing bad happened. Participants were randomized 1-to-1 to receive 2 doses of either the vaccine or placebo 20 days apart. So good. So this answers one more question here, that can I take a vaccine if I have some autoimmune disease? Yes. Can I take a vaccine if I have hepatitis C or hepatitis B or HIV, or I have diabetes or I have hypertension? So that is an important thing to keep in mind. Then primary efficacy endpoint. So one more thing that I would like us to keep in mind when we are looking through this document is that they have given a very interesting data point, which was thrilling for me to look at. That is the incidence of catching COVID-19 after the first dose. So what is the efficacy of one dose only? And that was a question here in a few seconds ago as well. Then what is the efficacy of the second dose and within the 7 days period? Because the way they are, the protocol is that, let's say this is the timeline: dose 1, then dose 2, 21 days apart. Then 7 days after is when they start calculating or observing if the disease COVID-19 occurred. So the question is, what about this time? What about this? And they have given data for this, and that is very, very interesting. So EDOG has a question that what about the people who are allergic to penicillin? So allergic to penicillin has two ways to look at it. Number one, you can say that they are folks who can be allergic to things. But at the same time, if this is specific to penicillin and they are generally fine, they're not hyper allergic, for example, pollen and the other things, and they have to carry the injections with them. So normally then they should be fine. But this is a more specific question that I think they would answer as you go for vaccination. So let's look at this now. So they're going to answer this question as well, that after first dose, what happens, after second dose, and within the 7-day period after the second dose, what happens here, after the first dose, what happens here. So keep an eye on that. That is an interesting question to answer, and they have the answer for that. Okay, so the primary endpoint was incidence per 1,000 person year. So they had so many people involved in the group that they could count in 1,000 person year the measurement. And then similarly, they had the incidence per 1,000 person year follow-up for with or without evidence of SARS-CoV-2. So check this out. They use the word with or without evidence of SARS-CoV-2. What they're talking about is that we want to see what is the incidence in people who already had the infection and then they got the vaccine, or they did not have the infection and got the vaccine, and then we are looking at them to have the infection or not. Probably you are maybe itching to find out, or all right, if I had the infection and then I got the vaccine, what is the outcome? Would it cause ADE, or would it cause allergic reaction and so on? So their data, they say it is not significant. The reason for that is that there was not enough people getting sick. But at the same time, then they say it shows that it is possible that the people who may get reinfected are going to benefit from the vaccination. So if you had the disease, then to prevent reinfection, if there is a chance of reinfection in you, then take the vaccine. So that means vaccine is fine to take after the infection as well. And so they have that data here. That means they tested it in trial. So continuing on, look at this. COVID-19 confirmed at least 14 days after dose 2. So that is one kind of criteria. Severe COVID-19, another criteria. And then CDC's definition of COVID-19 is the following, and they measured for that as well. So here they're telling us what kind of things they were measuring 7 days after, 14 days after, and what kind of definition of COVID-19 they looked at. Mild COVID-19, severe COVID-19, and then the CDC's defined COVID-19. With this, what are the side effects they looked at? And so here is a list of side effects: fever, cough, shortness of breath. Sorry, not side effects. These are the symptoms that indicated that somebody has SARS-CoV-2. So they were observing these symptoms. So continuing on, evaluation of the safety. So how did they evaluate for the safety? And this is what they did. For example, dose 1 is given, then 21 days apart, dose 2 is given. And then they are going to keep following up for 2 years. So they would keep seeing if there are any long-term effects. That's one. Then 7 days post dose 1. So today they have given the dose 1, and then they'll keep observing with more keen interest for 7 days after that. Then 7 days post dose 2, they would also keep looking at the person very, very carefully during that time. Then 1 month post dose 2. So then they would see that after the dose 2, a month later, how is the person doing? Then 6 months post dose 2, and then 2 years post dose 2. So they would keep following up with the vaccinated, with the vaccine group. Now, one more thing that was said today, I believe in FDA's approval committees, those folks who were on placebo during the vaccination will be given this information that they were on placebo and will be given a chance to become vaccinated as well. Okay, so continuing on, reactogenicity assessment. Reactogenicity assessment is the side effect assessment. So what were they looking for? Fever, fatigue, headache, chills, vomiting, diarrhea, new or worsened muscle pain, and new or worsened joint pains. This is what they were looking for. And then it's not that they were just asking for these questions. The participants could come back and offer unsolicited side effects as well, to say, I actually had this, or a joint pain, or I had something else as well. Okay, continuing. Of course, the disease itself, COVID-19 type symptoms, will not be considered side effects. They would consider the disease, and that is an important one, which is fine. Okay, so let's look at this. This is the phase 2-3 study safety analysis. So when they did the trial, the cutoff date was 14 November. That means they wanted to collect the safety data till 14 November. That means that they only used the population till 14th October. And then they collected safety data from them, and then they brought it into December. And that is how they kind of created the safety profile for a couple of months. That population, which is included by the 14th November for the safety data that has been provided, is called safety population in this document. There are still more participants who still need to be followed up and who still need to give data, so they're not included. Only those participants' data is included in the results that were able to give the data by 14th November. So that is the safety population. The folks who took part in it, 18 years and older, and then there were 16 years as well. Then here, check this one out. Phase 2-3 safety population, median follow-up time 2 months after vaccination number 2, comprised of a total of 37,586, greater than 16 years of age, enrolled by 9 October, and received at least one dose of study of the vaccine or placebo. So this is just some important parameters. Then check this out. HIV positive individuals were included in the all enrolled population, but not the phase 2-3 safety population, because the number of participants enrolled by October 9 was small, 120, and the median duration of safety follow-up was short. So what they're saying is they are participating in the trial, the number is small, and they're still following up on the safety. So I'm going to look at the questions as well. So ES says, so will the vaccine become like flu shots, once a year, one at a time, or just wing it until they get compliant or someone gets COVID and the spread starts again? So maybe I'm more optimistic. My thinking is that once we have the vaccine, some people are having the infection itself, right? So 10 million in the US. They are counted as someone who may have become protected. Some of them are not, maybe get reinfected, but majority of them are protected. Then another number of people are going to get vaccine. And that would provide immunity enough for 2 to 3 years. I keep going back to 2 to 3 years, which I have studied for SARS-CoV-1 and MERS-CoV. And cytotoxic T cell immunity can go on for 10 to 20 years as well. So my hope is, and I hope I am correct on this one, my hope is that once they give the vaccines or the person becomes infected, they become immune for 2 to 3 years. That will be sufficient time for this virus to go away from us. So Evo says, I got COVID, should I get the vaccine? According to this document, yes, so that if you have a chance of reinfection, vaccine would help bolster your immune system. And I'll show you those data points yet. There was a question I saw here, random wob. Question, what about kidney transplant patients? So I would consider it kidney transplant patient as an immune issue patient, right? Because they may be giving immunosuppressants to keep the graft in there. So if you see here, various immune disabilities were given the vaccine as well. What I do not know is, and it is not shown here, I went through the whole document word by word. I did not see anything related to transplant. So this may be a case to ask specifically from Pfizer or from FDA or CDC, but at least immune dysregulations or immune issues are covered here. Elderly and the healthcare people first. So Bob, are they recommending that those with known allergies, sensitivities, pre-medication with antihistamine prior to receiving the vaccine? I think they're saying that those with severe allergies don't get it. Okay, so Manpreet says, when will a normal human being receive a vaccine? So I hope we are all normal human beings, Manpreet. The vaccine has started in the UK. This is the vaccine we're talking about. And they have out, I would say, when are the general population outside the trial receiving the vaccine? That has started for 2-3 days and people are receiving the vaccines. And hopefully in the US, we would start that within next 2-3 days as well. Sunday is the CDC's meeting, and hopefully soon after that, we'll start it. Okay, so I'm going to go back to, so there's another, please do me a favor. When you have a question, maybe put the question mark or the word letter Q or the word question so I can catch the question. Dr. How and WHO would be following everyone for 2 years who has been vaccinated. So they have the data for everyone who's vaccinated, and they would keep asking. There are people who are not going to respond, or they're just not going to be there anymore, or stop the follow-up, but most of them will follow up, and so they'll keep doing it. EDOC says, will does with the vaccination be allowed to give blood? Very good question. So a vaccinated person will have some antibodies present in them. That is not a harm for anyone. They're almost convalescent plasma. So there should not be a problem. Morris says that question, has there been any safety issues with any vaccine several years being administered? There have been vaccines that have been stopped. But what I do not know is that several years later stopped. There are drugs that get several years later stopped because their effect on kidney or effect on carcinogenic effectivity is seen. But most of those are continuous usage drugs. Mackay Smith says, question, apparently it's on CDC website that vaccinated people are infectious to begin with. Please, could you do a video on that, as vaccinated people will need to isolate? No. Why would they say this? CDC website, vaccinated people are infectious? Are you saying contagious? Meaning they can spread the disease? They cannot. Well, what are they going to send? The spike proteins? And the spike proteins are not freely roaming around. Spike proteins are, think about it, spike proteins are manufactured in factories, that is cell, and then presented on top of the cell in specific complexes. So let's say this is a spike protein. It is presented on top of the cell as a immune presentation. So these spike proteins cannot go from my mouth to somebody else. And then what are they going to do there? That will be a strange thing if CDC says it. I would love to read to see what are they trying to tell. Okay, so Betty Rubble says, should we get titers after 1 to 2 years just to make sure we can mount a response? Good question. An important answer is this. When the B cells and T cells are produced, that are memory cells, they're going to sleep, and they would produce very low titers. So it is not necessary that the titers, the levels will be sufficient to give you comfort. The important thing is that if the infection occurs again, then these would wake up and work and the titers will increase. So yes, you can do that, but ideally you might see a very low level. Okay, so going back to the discussion, there are so many questions. Kweku Sam says, question, were there any participants that got pregnant? Yes, so we talk about pregnancy as well. There were the people who were pregnant before the vaccine started, they were excluded. But then there were people who did become pregnant during this vaccination. And the short of this is that there were, in placebo group, there were some abortions. In the vaccinated group, there was no side effects. But were there any side effects on the children? That is yet to be seen. Janet says that vaccine recipient became asymptomatic carrier at some point. Very good question. So they talk about it and they say, we are unable to tell if somebody who is vaccinated got asymptomatic infection. Of course, spike protein itself would not make you a carrier. But will it be possible that the virus came in and then the vaccine had created our immune system to take care of the virus, and then some small sample of the virus is still living on? Although I would think it's not possible, but let's say it is. What they said was, they said we had not done RT-PCR frequently to detect the asymptomatic patients. So they said we cannot talk about that. So very good question, and they have the answer in it. Machiavelli says, who selects the vaccination we get? I think that is going to be whatever vaccination brand is available near your hospitals or your doctors. Okay. So Peyton says, aren't allergens proteins? If so, how would a lipid cause an? So lipids can be allergens as well. The basic idea of an allergen is that it has to be a specific size. So lipids can be allergens too. Okay, back here. So this is the HIV, then the FDA's assessment, the safety data. So now we know the safety population, right? So up to 14th of November is the ones who would be referred in this document as safety population. Then here is the data that is starting, and this is where all the fun is. This is the general efficacy, and we know the general efficacy that we have 95% efficacy, correct? So I want to go beyond that because we've seen it. Here, disposition of all randomized participants. So here are the randomized participants. Vaccinated completed one dose was 99.8%. Completed two doses were 98.2%. So whatever the number that they had selected, they were 98.2% who actually completed the two doses, and in the placebo group 98.1%. So some of the folks were lost during the trial. They left the trial, or they just went away. Some withdrew from the study as well. So 180 in the vaccinated part had withdrawn. Of course, they did not know. They just simply said, I don't want to work with you in the trial anymore. I don't want to participate anymore. And 259 from the placebo had withdrawn. Adverse events. Look at this. Reasons for withdrawal. The people who withdrew, what did they say? 8 of them said in the vaccine group that, hey, I do not like the side effects. 5 in the placebo group. Death, 2 in the vaccine group died. 4 in the placebo group died. Although they will discuss the 2 who died in this group and discuss why did they die and when did they die. So we'll talk about it as well. Withdrawal by subject, 84 versus 157. Lost to follow-up, they just did not contact anymore. 80 in the vaccinated and 86 in the placebo. No longer meet eligibility 1 and 2. For example, got pregnant or like that. Refused further study procedures in the vaccinated 0 and in the placebo 1. So this is important. So this is again talking about the age 12 to 15. I'm going to leave this part and continue on. This is the demographic characteristic. So if you wanted to see that how many men, how many women, then what was their mean age, mean year, how many people from 16 to 18. So look at this, 15, then 16 to 55, 11,589, and so on. So there is a decent amount of data here. If you can see American Indian, or Alaskan Native, Asian, Black or African American, Native Hawaiian, white, multiracial, not reported, Hispanic or Latino, not Hispanic or Latino, not reported, comorbidities, yes, no, obesity, and they have the numbers for them. Decent numbers, good numbers to look at. So then I want to go to those things which are useful for us to continue to understand this. So look at this. 4 participants without evidence of SARS-CoV-2 infection prior to 7 days after dose 2. So dose 2, where is my... So dose 2, so if I take this out, what are they talking about? They are talking about this group. This is the group that is after dose 2, but 7 days before. Because after 7 days, they start measuring. So this is still a window that they are not measuring. So look at this data here. For participants without evidence of SARS-CoV-2 infection prior to 7 days after dose 2, vaccine efficacy against confirmed COVID-19 occurring at least 7 days after dose 2 was 95%. So this is 95% we know. The case split was 8 in COVID-19. This is that reported data. And compared to 162 in placebo. So that is good. We like it. And then look at this. The 95% credible interval for the vaccine efficacy was 90.3% to 90%. So that is the window, because it is statistical data. So 95% is what we say, but ideally anywhere from 90.3 to 97.6, which still is all good. Okay, so then for participants with and without evidence of SARS-CoV-2 infection before and during vaccination regime. So now this is before the vaccination and during the vaccination, with or without infection. So those people who had the infection before and got vaccinated, or those who had dose 1 and got vaccinated, or those who had dose 2 and got vaccinated but 7 days before the countdown started, at least 7 days after dose 2, was 94.6%. So what are they talking about? They're talking about this. They're saying anyone who had COVID here before the vaccine and then they got the vaccine, or they had the vaccine and then they got COVID, or they had the vaccine dose 2 and then they got COVID, but before the 7th day started, where the actual countdown, protocol countdown started. In these folks as well, the efficacy overall was 94.5%. So that is good. Now, the 95% credible interval for the vaccine efficacy was 89 to 97. So in these folks, the range only dropped to 89.9 to 97. So that still is a good range. So including those people who actually did become infected or had infection before and then got the vaccine, even when they got the vaccine, the results were still very good. Okay, so continuing on. So if you see here, this is the efficacy of the vaccine against confirmed COVID-19 from 7 days after dose 2 in participants with and without evidence of prior SARS-CoV. This is very important. People who got vaccinated and had the infection before, and people who got vaccinated and didn't have the infection before, collectively talking about all of them, the efficacy, if you see here, was all participant, look at this line, 94.6%. So those who were infected and got vaccine also benefited. So that is excellent. Then is the subgroup. So now they are going to talk about various groups and their specific conditions. So this is a very important point here. Vaccine efficacy point estimates were uniformly high across the subgroups examined, with the exception of participants identifying as multiracials and participants with evidence of prior SARS-CoV-2 infection at enrollment. And do you know why? Because they didn't get enough infections to be able to collect that data and say they got infected or not, which is probably for this previous SARS-CoV-2 infection, it is a good thing. So what they're saying is, if you had the infection before, then you got vaccinated, there was very limited chance that you would get infection again. So here, here is that data breakdown. Again, very thorough and very good breakdown is available. What I wanted to show you is this part. Look at this baseline SARS-CoV-2 status. Those who were positive previously and then they were given the vaccine. One here and then one here. And those who were negative, 8 and 164. So I do not know what does this really mean, that they were negative before and then they were given the vaccine. That should be the case for everyone. Or were they positive before and then they had become negative at the time of vaccine and then they were receiving the vaccine? So this one line, even when I read their subtext over here, negative N-binding antibody result at visit 1. So negative neutralizing antibody result at visit 1 should be for almost everyone who is not infected. So I didn't understand. Probably I need to become a little smarter for this one. Okay, so I'm sure I'm missing something, and something simple. Then I want to continue on to this discussion. Severe COVID-19 cases. This is really important. So in the final analysis of evaluable efficacy population, our 4 participants had severe cases. So in there, 1 subject who received the vaccine and 3 who had received the placebo. So total 4 severe cases, 1 in the vaccine group and 3 in the placebo. But check out the definition of severe. I was kind of happy about that. Here is what they're saying. The vaccine recipient who had severe COVID-19 disease, the person who had the vaccine and developed the severe disease, met the severe case definition because oxygen saturation at the COVID-19 illness visit was 93% on room air. So of course, we have treated patients below that at the room air. So this is correct that this is severe, but this is really not that severe. The subject was not hospitalized, did not seek further medical care, and did not have risk factor for severe disease. The 3 placebo recipients who had severe COVID-19 disease met the severe case definition of the following reason. 1 subject had an oxygen saturation of 92%. Again, not a, in the given context, not a huge issue. Without other severe disease criteria, 1 subject was hospitalized, so this is severe, for non-invasive positive pressure ventilation with bilateral pneumonia, and 1 subject had an oxygen saturation of 92% and ICU admission for heart block. So these were placebo. There was just 1 93% room air. So there is, Janet is kind of amused. Nowadays, look, I have seen now so many folks below 93% at room air that this seems to be okay, but that is their cutoff point. So there is an interesting question I just saw. I missed the questions. So here is a question, Eva Johnson. If there were to be an age range for the vaccine, would the size of the person change them being able to get vaccinated? Like if they were old enough to get it but are short, what would the rules be? So they're giving it to healthcare workers and older population first, regardless of whatever. So if they're short, the other tiers will not get it. Okay, so back here, we're almost near the end of it, and then we can have more discussion here. So that was the severe cases, 1 in the vaccinated, 3 in the placebo, but please see the definition of severe here. Then additional efficacy analysis. This is very interesting. I loved this one. So check this one out. First COVID-19 occurrence after the first dose. So if you say that, hey, if I got my first dose, and I think somebody asked that question here as well, I believe Vincent, I got the first dose and now I'm not going for the next one, or I'm traveling or something happened. Overall efficacy was 82%. So efficacy drops. So that means the first dose is not as effective. But if you go with the definition of FDA and CDC that had said vaccine should be at least 60% effective, so then here 82%. After dose 1 and before dose 2. So that was the overall efficacy. Now they're giving specific parts. After dose 1, before dose 2, 39 people got COVID-19. This is an interesting data point. And 82 in placebo, the efficacy has dropped to 52.4%. So not very effective. So it reduces the chances to almost half and half. So after dose 1, the effectivity does not rise to a point where you can say, I am safe. You still have your chances become half of getting the infection, but you can still get it more than after getting 2 doses. Dose 2 to 7 days after dose 2. So this is, so they were talking about, so if I go back here, first they said this group, the efficacy in this group is 52%. So not good. Now they're going to talk about efficacy in this window. So this efficacy is 90.5%. And then from here onwards is about 95%. This is very important. So I hope that I know I'm repeating it, but this question had been asked so many times that, hey, what about one vaccine only? So one vaccine dose only, 52%. What about the 7 days after the second vaccine? Am I still at risk, and how much risk? Not too much, 90.5, not bad. And then after the 7 days, 95%. Then there is another question, I answered that yesterday. Why this change? Because here we are starting the immune system to become ready. Here we are kind of boosting it, and then it learns its lesson and becomes ready really over here. What does that ready means? Here is one B cell. Let's say this is a naive B cell. Then we trigger it and we say, hey, B cell, wake up, learn to be making the antibodies. He looks like blind to me. Make antibodies. And it starts making antibodies. And not only it makes antibodies, it makes copies of itself, which is called proliferation. So let's say you give the first dose and we have proliferation, or the number of B cells produced are, I'm just going to make up some numbers to explain it, maybe 1,000. It's in millions, but just to understand this. 1,000, number one. Number two, the B cells conformation for the, or binding capacity, binding affinity is not very well. So maybe it is 70%, I'm making up a number here. Maybe it is 70% good at binding the antigen. If you give it another dose, these B cells, they would proliferate even more. So here they were, and now they would make even more B cells, because they're going to say, oh my God, the antigen is serious. It has arrived in the body again. So we need to increase our number. Number one. Number two, they are going to do affinity maturation, which means that the new antigen that came in again, that is going to make them a little more tighter in their work with the antigen. So now they are more specific, even more specific, which we call in medicine affinity maturation. Their affinity has matured, and they're more in number. Both of those things are very, very useful for a better response. So you can say that affinity maturation occurs here even more, plus the proliferation even more occurs here, which causes this area to become very, very safe. So that is the basic answer to this one. So I love this one. Continuing on. So here they're just talking about the same thing that we just saw. Then adverse effects. So here, let's look at the normal adverse effects, then the Bell's palsy, then the death. So let's look at all of them. And then they'll talk about pregnancy as well. So look at this. Adverse effects. They were looking at adverse effects after dose 1, dose 2, and so on. So if you see here, smaller numbers, 0.4% after dose 1, 0.2% after dose 2. Within 30 minutes of the dose, somebody says, oh man, I have a lot of pain at my injection site. So after immediately giving the dose and within 30 minutes window, 0.4% in the after dose 1 and 0.2% after dose 2 in placebo. In the vaccinated, 0.4% and 0.3%, so no difference really. Both of them complained the same way. After 7 days, in the vaccinated group, after 7 days or during that 7 days period of getting the first dose, 78% had some kind of a complaint. In the placebo, 12%. So that is a significant difference. After dose 2, 73% had a complaint of some sort, and then 10% in placebo. So once again, significant. So this is an interesting data point as well. And now what were the reactions? Pain, redness, swelling was the local reaction. Where the injection was given, there was pain or redness or swelling. And you can see the numbers here. Then frequency of solicited local reactions within 7 days after each vaccination. And this was, I think, greater than 50 years of age. So again, the percentage is here 71%, 55%, redness was 4%, swelling was 6%. So this is local reaction. Now, systemic reaction. Look at this. The frequency and severity of systemic after effects were higher in the younger than the older ones. Isn't that interesting? So younger population actually reacted more, and they had more after effects than the older population. For both age group, fatigue, headache, and new worsened muscle pain were the most common. So the most common thing that you can probably expect is fatigue, headache, and muscle pains. So they have given that data as well over here. Then this is the serious side effects that were, non-serious side effects that were told by the patients. So what are these? Number one, lymphadenopathy. So they say, I think in one person who received the vaccine, their lymph nodes swelled up. And that can happen. What happens is when we have some antigen in our body, so let's say this is the antigen is picked up by macrophages and neutrophils and other things, other cells, innate arm cells. They kind of pick them up, and macrophages would now present them on their surface, correct? And then they would enter the lymphatics and run to the lymph nodes. This is like we arrest someone and go to the police station. The lymph nodes are like the police stations. And over there, B cells live there and T cells live there and other cells live there, for example, follicular dendritic cells and so on. So these guys would run there and go and say, hey, I caught an antigen. Look, I have an antigen here. And so they would present that antigen here. When they would present the antigen, the B cells and T cell who would look at the antigen in turn become proliferated. They would increase in number. And because this is happening inside a lymph node, and lymph node has a capsule around it, a tissue capsule, and they cannot really become enlarged easily, they become painful. This is what lymphadenopathy is. So lymphadenopathy can occur after an injection of some antigen. Not a big deal. Some of you may have actually experienced that after some infection, for example, a wound on the leg or arm causes some lymph nodes to swell up for a few days. So lymphadenopathy was seen. How many people? More in the vaccine group, which makes sense, 64 people, and the placebo group 6. So I said 1. So 64 versus 6. And again, I'm not too worried about that. It is actually possible. Bell's palsy. So Bell's palsy, somebody had asked me on Twitter today, Bell's palsy. There were 4 cases of Bell's palsy in the vaccination group. So Bell's palsy was reported by 4 vaccine participants and none in the placebo, so 0 in the placebo. These cases occurred at 3, 9, 37, and 48 days after the vaccination. 1 case onset at 3 days post-vaccination was reported as resolved with sequelae within 3 days after onset. So most of them, I think except 1, everybody had resolved, and I think 1 was still reporting their status. Now this is important. The observed frequency of reported Bell's palsy in the vaccine group is consistent with the expected background rate in general population. Although my answer to them was, number one, yes, I understand, it's fine. It got resolved as well. And my counter statement to my own statement will be, well, if that is the case, then placebo group should show a similar number because this is the general population rate. So ideally, placebo should show the same number. FDA has not felt that this is a big enough of a problem because they got resolved very fast. So what FDA said was, we understand what is going on, and we would ask that when people get the vaccine, we just keep looking at Bell's palsy to make sure that it is transient if it occurs and it goes away. So that is a Bell's palsy. Continuing on, we are at the end now. Deaths. A total of 6. 2 deaths in vaccinated group and 4 deaths in placebo group occurred. Both vaccine recipients were greater than 55 years of age. One experienced a cardiac arrest 62 days after vaccine number 2. So 62 days after, so this was the second dose, and then about 62 days after, one death occurred here. So and died 3 days later. So 62 days after vaccination number 2. So he developed cardiac arrest, and then 3 days after he died. And the other died from arteriosclerosis 3 days after vaccination number 1. So the other person, after the vaccination number 1, 3 days after, he developed arteriosclerosis. Arteriosclerosis means hardening of the blood vessels. I don't think that this vaccine can cause hardening of the blood vessel to an extent that the person actually dies of it. So he may have a condition. The placebo recipient died from myocardial infarction, hemorrhagic stroke, unknown causes. 3 of the 4 deaths occurred in the older age group. Greater than all deaths represent events that occur in the general population of the age groups where they occurred at a similar rate. Then is the non-fatal serious side effects. So what were these side effects? So some people had appendicitis, not related to the vaccine, but anyways, appendicitis. Appendicitis was reported as severe after effect in 12 participants and numerically higher in the vaccine. 8 on vaccinated side and 4 on the other side. All of the vaccine participants and 2 placebo participants were younger than 65. So that is appendicitis. Then they said there's another lymphadenopathy. So they have an FDA's opinion over here. And that opinion is that by giving the vaccine, it is possible that we will cause the lymphs to swell up. So it's okay. Now, suspected COVID-19 cases. That is an interesting one, that some people actually got the COVID-19. So 2 serious cases of suspected but unconfirmed COVID-19 were reported, both in the vaccine group, and narrative were reviewed. In one case, so this is very interesting, vaccine group, severe COVID-19 cases reported. So let's see what happened. In one case, a 36-year-old male with no medical comorbidities experienced fever, malaise, nausea, headache, and myalgia beginning on the day of the dose 2 and was hospitalized 3 days later for further evaluation of apparent infiltrates on chest radiograph and treatment of dehydration. So he had chest changes, he had symptoms like COVID, and he had dehydration. RT-PCR was negative, and a chest CT was reported as normal. The participant was discharged from the hospital 2 days after the admission with chest imaging findings that are difficult to reconcile. It is possible that this event represented reactogenicity. So they're saying maybe it was vaccine's reaction, or maybe it was some other disease which looked like COVID. In the other case, so this was one person, right? In the other case, 66-year-old male with no medical comorbidities experienced fever, myalgias, and shortness of breath beginning 28 days post-dose 2, 28 days after, and was hospitalized 1 day later with abnormal chest CT showing a small left-sided consolidation. He was discharged from the hospital 2 days later, and multiple nasopharyngeal PCR tests collected over a 10-day period beginning 2 days after the symptom onset were negative. So it is possible that, though highly unlikely, that this event represented a COVID-19 case with multiple false negative tests that occur more than 7 days after the completion of the vaccination, and more likely it represents an unrelated infection. So they're saying it is kind of unlikely that so many PCRs were negative, but maybe, or it may be another infection. So that is interesting. This paragraph is very interesting as well. Are you guys holding up fine? So I'm going to come back to that. Just one more discussion here for the pregnancies, and then I'll come back here to the live stream. So check this paragraph. Among 3,410 total cases of suspected but unconfirmed COVID-19 in the overall study. So 40,000-ish people study, there were 3,410 cases of unconfirmed, but they looked like COVID. 1,594 occurred in the vaccine group, 1,594, and 1,816 in the placebo group. So almost equal. Suspected COVID-19 cases that occurred within 7 days after any vaccination were 409 in the vaccine group versus 287 in the placebo group. So what they're saying is within this period of time, there were 409 people in vaccination group that presented with the symptoms of COVID-19. Now, it is possible that the imbalance in suspected COVID-19 cases occurring in the 7 days post-vaccination represent vaccine reactogenicity with symptoms that overlap with those of COVID-19. So they are saying it is possible that this spike protein created some symptoms that seemed like COVID-19. And so that was just a reaction to the antigen. Overall, though, these data do not raise the concern that the protocol-specific reporting of suspected but unconfirmed COVID-19 cases could have masked clinical significance. So they are saying that this data is not masking any severe side effects or some side effects that are not taken in the name of the protocol. So meaning not a big problem. And then pregnancies. Female study participants of childbearing potential were screened for pregnancy prior to each vaccination with a positive test resulting in exclusion or discontinuation from study vaccination. The study is collecting outcomes for all reported pregnancies that occurred after the vaccination or before vaccination and not detected by pre-vaccination screening. So what was that? 23 pregnancies were reported through the data cutoff date of 14 November. 12 in vaccinated group and 11 in placebo group. Study vaccination occurred prior to the last menstrual period in 5 participants, 4 vaccines, 2 placebo, within 30 days after the last menstrual period in 8, and 4 and 6, and then 30 days after LMP in 1 participant. So unsolicited after effects related to pregnancy included spontaneous abortion and retained products of conception, both in the placebo group. Pregnancy outcomes are still to be known. So nobody who was pregnant, who became pregnant after receiving a dose of vaccine, or whose pregnancy test was missed and received the dose of vaccine, they did not have any side effect. And in the placebo group, unfortunately, there was an abortion and a missed conception content in the uterus. And what happened with the children will be seen when they are born. In my opinion, what would happen to the children, I've seen pregnant women who got COVID, and the IgG goes to child as well, and the child is born with the IgG, which is a good thing. So I don't see any iatrogenic effect, that is any child damage effect, but they still have to look at the data and tell us. So this is what we have in that area. So I wanted to make sure that we go over it because our answers, many of our answers are there. Can I just look at a few questions here and then we stop? So Kent Maurice says, is it possible that a vaccine can cause a PCR positive test for SARS-2 if one is to get infected after the vaccine? Does the PCR test differentiate between the actual SARS-2 virus and the vaccine? Yes. So the injection is given in the muscle, picked up by the immune cells and muscle cells. That mRNA from the lipid will have to travel all the way here to the nasopharyngeal area, then divide and increase to become significant for PCR. So that's not possible. So Venki says, how is the chimpanzee virus managing to enter the human cell using the newer vaccine? So that is the Oxford vaccine or Sinovac vaccine, traditional vaccines. Let's talk about that separately. I want to talk about these as well, but it would just muddy up this lecture. These are called weakened viruses. They still have the capability of entering, but they do not have replication capability. Sneaky Pete says, is Q is a fool, and if you really believe in Q, you need to okay, so this is some discussion going on. M. Gregory says, question, do vegans have less symptoms of COVID? I am not aware of this. That's a good question. So any hot question here that is still left out after reading this? EDOC says, are the people tested for illness, medication, or relying on what the volunteers only tell them? So there are both kind of things. The solicited after effects are where they ask them questions, and unsolicited is where the people just volunteer that, hey, I got this. I'm seeing if there are more questions. So it seems like there may not be. So there is a question here. Ed Monher, a question, what are the odds of side effects years later? So if you think about it, the side effect that can continue years later can only be, in this case, if the immune system has become dysregulated and continue to damage us like long haulers. And that, I think, with the vaccine is not going to be possible because there is no replication, continuing replication to bother the immune system, plus the dosage are set in a way that they do not trigger the immune system to become dysregulated. There has been no cytokine storm in these people, the one who were participating. So in my opinion, in my understanding, this should not happen. Falcon Fly High says, "Can you please reply to me, please? I'm concerned about it." Falcon, what is your question? Okay, when someone cardiac arrests means there is a chance of that. Will you agree? When someone cardiac arrested means there's a chance of that. Will you agree? So that cardiac arrest case was 63 days after the second dose, where they developed cardiac arrest and then they died 3 days later. But that was not, I mean, from just this data, it shows that it was not related to the vaccine. It was 63 days after the second dose. So 2 months and something after the second dose. And what should the vaccine do to cause cardiac arrest? I don't think that that is related. Bob says, thank you for your review. Do you have similar documentation for the Moderna vaccine, and will you be reviewing that as well? I believe the FDA will be evaluating next week. Yes. So as Moderna's vaccine comes in, we would review that as well. As uh, other vaccines come in, for example, Oxford or Sinovac, if they come in, we'll review those too. Steve says that did you watch any of the FDA meeting today? And if yes, what did you think? I watched it. It was long meeting. I kept it on my phone, and just the whole day it kept going. I thought, when would it end? Um, man, it was interesting. I could not. Sometimes I could not understand. For example, they argued about 16 to 18 years and should they give them the vaccine or not. And one population group, the doctor said, no, we should not give them the vaccine. It should be for higher age groups, and there is no side effects, no much effect in the younger age group. Then another set of doctors said, well, don't prevent from giving it, maybe don't prioritize it, but at least don't say don't give it, because then if a doctor administers it, they would be banished, or they would be said that you've done something out of the protocol, like ivermectin and hydroxychloroquine. This is my part of the comment. So I was seeing these kind of discussions, which to me didn't make any sense. Yes, make it 16, make it open from 16 and onwards, which they finally did. But they had these kind of discussions. I saw some of the comments from public as well, some concerned, some saying, hey, too rapid, too fast, not fully tested. And so I had been reading up so much about these things that I was kind of, these seemed like um, normal things to me and not too concerning, but it was interesting. So Partha has a question. Is it possible to change the vaccine type from mRNA to DNA or attenuated vaccine in future, or it should be the same? I would prefer mRNA. mRNA stays outside this nucleus, doesn't go into the DNA, doesn't do anything there, just goes to ribosome, makes the antigen, and gets done. Why should we change it to DNA? Gene says, aren't all allergens proteins? So I think I talked about it before. 1x2 says, Doctor, when are you going to comment on Dr. Corey's Senate statement? We can comment any day. So the problem is this. There is no outcome. Doctors are still not giving ivermectin. I usually don't discuss this, but you know that there are complaints about my company from doctors now, and they have started various ways to shut us down because they do not like that we talked about off-label drugs. And I am having to work with those things at this time. And I was thinking today that maybe when the pandemic is over, Drbeen is also over and it is shut down as well. And that is the outcome of this whole thing. And I'm not ashamed of the work I've done. But the point I'm making is that even with all of these, I hope that that registers, and I hope that NIH or others who, when they don't approve something, they create a taboo, they can approve it. Otherwise, in the bigger society, medical society, it is still looked down upon to talk about hydroxys or zincs or ivermectin. Media has been more than dishonest. And this, I would say, from any media. If you go to Fox, they have their leaning, and over there, they close their eyes to the other side of the leanings. If you go to CNN or MSNBCs, they have their leaning, and they close their eyes to the other side. And they all are sitting in their echo chambers, and they're causing damage in their own ways. But what I dislike about media's coverage is that without actually looking, either don't talk about something, or if you're going to talk about it, then do your research. So calling Dr. Corey as a fringe group who is promoting a anti-lice drug. So using such words that when you listen to them, you already develop a negative image. That is not journalism. That is maybe punditism, but our media and punditry has just become so combined together that it is a difficult thing. So you started me on this Dr. Corey's statement. I wish it makes a difference, but I see the community, medical community, the medical leadership to just stay stuck. And why? Look at this. Giuliani became sick. He went to hospital and 2 days out, he's back out. Why? I think they gave him the same cocktail they gave to the president. A very good friend of mine, when I used to be in Massachusetts, my wife used to run a restaurant there, and he used to come there, and I used to be there as well, and we used to talk. And he was a teacher, I believe, in Brown University. Very, very decent human being. He's on a ventilator, and it is difficult to come back from the ventilator. And his wife told us a couple of days ago. And I keep thinking that what kind of a society are we, that we know there are solutions? Fine, forget about hydroxy and ivermectin. We know that Regeneron helps. We know Regeneron, dexamethasone, and remdesivir combined. Remdesivir not too much, but still combined, it helps. Regeneron's cocktail helps. And we are such a cruel society at this time. And again, I'm talking about the leadership, that they use it where the people are big guns and they're letting others slip away. So ivermectin or Dr. Corey, I don't think has any. I have never been so disappointed. I have always been upbeat. That is why I've done these 235 lectures now. But I am so disappointed. I was telling my wife today, if I went on a ventilator or if I became severely ill, they're not going to give me Regeneron. They're not going to do what they did to Trump and brought him out in 2 days, or Giuliani and brought him out in 2 days. They're going to let me go on a ventilator and die and just make me another number. This is how bad the medical community is. And media is just, it's BS at this time. So I wish they listened to Corey's and Dr. Merrick's discussion. I wish that they make it available for humans' sake, not for Republican sake or Democrat sake, or which medical community is right and which pharmaceuticals is going to profit. I wish they'd stop that. They're not stopping it. Okay, so Amber's adulty life. When will those who are 65 plus and with weak immune system be able to get the vaccine? So I think that they're starting with the nursing home residents, then they're starting with the essential workers, and then they are going to the uh, people at risk. So if this is somebody who is already in a nursing home, then they would get the vaccine in the first priority. But if they are at home and they are at risk, then they are in the phase 1C of the first phase. Now, when does that become engaged? It depends upon the doses of the vaccine. So it can go on till June of next year. So my teacher here, Professor Mulazim Hussain Bukhari, Dr. Mubin, do not disappoint from this, as we are doing a job for humanity. We are not working for any politics. That is correct, sir. But at the end of the day, there are certain levers that are only in the hands of politicians or the medical leaders. For example, ivermectin is not becoming allowed. Somebody was talking with me yesterday, a doctor, and saying that I'm seeing my friends in New York who are prescribing ivermectin, and their prescription of ivermectin is denied at the pharmacy. Who has the nerve, or who should have the nerve in the pharmacy to deny a doctor's prescription? And here we are, that they're denying a doctor's prescription at the pharmacy. And so that means if somebody above the doctors have to come back and said, this is okay. So Corey's discussion, Dr. Corey's discussion, or the Senate hearing, what is the outcome? So there's a question here, Rishi Kisan. Dr. Bean, would you recommend these vaccines for people with allergic rhinitis and chronic urticaria? Yeah, so they used them with autoimmune disease patients, and they were fine. With the lymphadenopathy regional in the drainage region of the injection, correct. So that was in the axillary region, which was kind of related to the injection. Cool. So this is the discussion. So I says your voice was inaudible there. What did he say happened at the pharmacy? In New York, some pharmacies are just simply denying, rejecting ivermectin prescription. So a friend doctor said that his friend doctor, so he was talking with me and he said, are you experiencing anyone here getting your ivermectin rejected? And I said, in my group, nobody is giving it, and I have not heard rejection yet. And he said that my friends, and he's a very good friend of mine, he said, my friends' ivermectin prescriptions are being rejected at the pharmacy. They're not filling it. This is like we have caged the Americans, and then we are letting them die. And we are not letting them protect themselves. Even that is disallowed. Arshad Iqbal says, question, any data on using ivermectin in nursing home residents? Um, I do not know any specific study, but it is, ideally it is safe. Rajesh says, Dr. Bean, you are on strong scientific footing and your videos vested interest won't succeed against you. I hope so. There are some complaints that have happened and there are some folks who are looking into us at this time. But I said to my team members, there is nothing, there is no cheating we have done. There is nothing wrong that we have done. So uh, anybody can come and look as much as they want. But this just tells me that there are folks out there who, for their own ego, are not even able to give a chance for a safe drug to be tested. They would go and tear down anything that is related to it or that is offering that, just out of anger. I do not know, people are so full of pain that they would go out of their anger and do these things. Okay, so Tim says, is it possible for it to affect wrong for a hemophilia patient? So hemophilia patients, again, it is an immune system trigger, and you get hemophilia patient get infections or they get cold and it is fine. So ideally this should be okay, but you should talk with your doctor, because I'm sure that Pfizer and others would give specific instructions in these cases as well. Ed Moner says, question, would you say that the increasing cases are from people not wearing masks? There is so many. We just have such a huge number of people who are now spreading. If we could spread from 1 to 10 million, imagine if we have 10 million or active, let's say 500,000, they are going to spread a lot. Yes, wearing mask is very important. Social distancing is very important. Hand washing is very important. But it's just that the basic core, just like the viral load, it is the human load of infected human is so large that society cannot just escape from it at this time. So last couple of questions and then we stop. AD says, will the vaccine cause spike proteins to mimic ACE inhibitors? No. So I have explained this many times and I'll say it again. Vaccine is going to make spike proteins inside the cell, and cell will pick them up and present them on MHC1 or 2. So spike proteins are not being secreted by the cell. So these spike proteins are not going to go anywhere to get attached to the ACE2. Yes, that cell that would be killed by natural killer cells may spill some of the spike proteins, but these will be normally, just like with other damaged tissue, macrophages and neutrophil are going to pick them up and present them on antigen 2, MHC2. So no, they're not going to act like ACE blockers. Yeah, Doddy says, will the vaccine be forced on us, as the vaccine could change our DNA? Yes or no? Number one, I've explained it many, many times. It's not going to change our DNA. An RNA vaccine cannot change our DNA. Number one. Number two, I don't think it will be forced on us. I don't think that anyone can come in and say, everyone line up, I'm going to inject you. So I don't think that is going to happen. But that, I mean, future would tell us. This is my thought. So let's stop it here. We have, I thought this would be 30 minutes today, but we have gone on for 1 hour and 39 minutes. I hope it was useful and it was valuable for you. Please like, subscribe, and share. If you wanted to support the work that we are doing, there is a link in the description. You can use that to support it. And then I would see you tomorrow. Bye-bye.